to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 25th episode of the Weekly Bioanalysis. My name is Dominic Guarino, and I am the Senior Scientific Advisor for all things related to ligand-binding assays, biomarkers, and flow cytometry. I have been with KCAS for over 7.5 years, and I've been in the bioanalytical space for close to 26 years. I'm here with my co-host, John Perkins.
Hello, I'm John Perkins, Senior Scientific Advisor with a focus on LC-MS technologies. I've been with KCS for about a year, but I've been in the bioanalytical space for around 26 years.
Today's podcast, as always, is brought to you by KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical laboratory located in Kansas City, Kansas, serving the pharmaceutical and biotech industries for over 40 years now. John and I are senior scientific advisors at KCAS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCS services. We are glad that you are tuning in to us today. As we mentioned earlier, this is our 25th podcast recording. It's our silver anniversary. But if you are interested in any previous podcasts, they can be found at kcsbio.com. And you can also follow us on Twitter at The Weekly Bio and at KCS Bio. So if you are new to the podcast, I'm located in Kansas City. John is in upstate New York, and our producer, Jeremy, is in Missouri. This podcast, we hope, gives us a chance to connect with all of you, and we hope it gives you a chance to get to know us. And even more importantly, it's a chance for you to get to know KCS and the services we provide. As always, the podcast will be a review of the latest news and resources, and then a focus of a topic of our choosing before discussing any feedback we've had from our audience. As always, we're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. Then we will wrap up and give you a little teaser for next week, letting you know what is coming in the future. So again, we're very excited to have you join us on our very special 25th Silver Anniversary podcast. We hope we have a fun episode, but to kick us off, John's going to go over our podcast topics a bit.
So, as usual, we're going to touch on the news first, looking at COVID items and then non-COVID news, all related to the drug development field. And for our main portion, we're going to do something slightly different. This is actually we thought, as it's the 25th episode, let's maybe cycle back and give some information as to how we ended up where we are today, both Dominic and I. So it's talk about our degrees, our careers, and what shape to then take on the senior scientific advisor role at KCAS. Let's move to the news, and obviously we, as usual, will start with COVID topics. First up, there's another mRNA vaccine candidate coming through, successfully completed animal studies in mice and monkey. This is Sanofi partnering with a company called Translate. I mean, the mRNA is the similar approach to Pfizer and Moderna, so that same method of delivering the vaccine. But it's also like following a 2-dose regimen similar to the Pfizer and Moderna vaccines. But according to the article that I looked at, the animals, both mice and macaques, developed neutralizing antibodies and an T-cell response. So they're also looking to they're not just looking to run. They also have challenge trials ongoing where animals are actually infected with the virus directly and then seeing the effect of the vaccine on that. Any comment on that?
Yeah. No, John, you did a great job of going over that. I'll just share that they seem to have some real strong evidence to show T helper 1 and T helper 2 responses, which is some much more detail than normal in these types of postings, which I think the other vaccines in this space have shown that type of efficacy, but they came out of the gate with it, which is good. I think there's somewhat of a roadmap being laid for this organization here, but that looks very promising, and we love more vaccines and pretty hopeful with this sort of candidate. Yeah.
Yeah, it's nice to see no one's resting on their laurels, and there's obviously ongoing research that we have the first batch of vaccine candidates, but we know there's more behind them in case the current front leaders stumble. So the list is obviously growing. At some point, I need to revisit the endpoints listing to see where they rank everyone in terms of top 38.
I think you have a podcast number 29 topic for us, John. I think that'd be a great one, just to revisit the vaccines and therapeutics. But I feel like we've talked about that one. We're about due for it, but that would be a great podcast. Yeah.
what I think is the bigger news, and I still not really sure how I feel about this. There's been a lot of talk about this, particularly in the UK, about the use, moving on from the challenge trials, which we're saying, Sanofi are doing for animals, but actually doing challenge trials in humans, where the plan is to get 100 volunteers, 18 to 30, and intentionally infect them with COVID. First leg is to expose them to rising doses of the virus to see how much virus is needed to cause infection. And then the second step is to look at testing various vaccine candidates against the virus with this approach. I mean, it's like, I shall let you comment, then I'm going to swing back in with my thoughts.
I'm okay with it. I think this is the logical step. Is it 6 months sooner than I ever dreamed it to be? Yes. Does that mean it's harmful and rushed? No. The reason why this goes quicker is we're moving some of the bureaucracy rather than the science as well, John. So I think I'm okay with it.
So I think my concern comes from the science, actually. So the talk can launch in early 2021, but I mean, we're still at the stage, been discussing over multiple weeks, that we're still digging up articles where people are saying, oh, here's a fresh mechanism of action of this disease. Here's another way it's affecting the various organs. We don't really know what the effect of this disease is on people yet. We haven't tied that down. There isn't really complete understanding. I would not be volunteering for this. And then looking at the structure of the design of the study makes perfect sense. But when they actually talk about, oh, we're going to expose them to the virus, and once we've infected them, then we're going to administer remdesivir to counter that. But remdesivir, we're still seeing mixed data from that in terms of clinical trials as to whether it's even effective or not. I know it's had emergency use authorization. Remdesivir, I don't think it's not convincing yet that it's absolutely a therapeutic for this. I may be speaking a little bit out of turn, but to actually infect people with this and then say, oh, we're going to give you something that has been approved, but it is not a cure. It's really not. We can't guarantee it's going to fix you, to something that could actually then have a real deleterious effect on these volunteers' lives. I think it's early and irresponsible, to be honest.
Well, I'm going to make the assumption that what they're challenging them with, John, is not the same as like an active infection in a patient. So I understand your concern there. The way I see this is like in the allergy space, when people get peanut allergens given to them and things like that. So I don't know enough about how they're doing this, but it's not the same as like, a sick person coughing on you in the bus, right? Like that's not what's happening. Or like, even to the level of exchanging bodily fluids in any sort of manner. So,
Oh no, I understand that. I mean, I don't know how they're doing it, but yeah, well, we'll see. I mean, obviously,
so that's why I think if I shine that lens on it for you a little bit, do you have less of a concern though? Because I don't think they're given like, big doses of, I think they're given like proteins of the targets to sort of challenge them. And why they give remdesivir, I think, is probably more of a safety at this stage. And you're right, it's TBD, but if they can prove it at this stage that it works, then you can just give it safely. And it does, it has a window, is how I describe it to people. I think remdesivir, hydroxychloroquine, even, and I don't want to. I think I just heard John might have perked up a little bit, like but no, like those are all things that like have maybe some benefit in a small window of time. That's the key thing. And remember, we talk about how much the bolus of the infection you get. All of that is going to impact those drugs. And then your own immune system, it has a subpopulation and a small window of efficacy. Again, I think in this case, it's more just a safety thing, like a check the box. You brought some great points. I hope I countered some of them and we could have a great debate about this. But I think there is, in my mind, if I boil it down to like getting, having your kid go in and get challenged with peanut allergens, which by the way, there are still, percentages of the population, a high percentage that would never let their children do that. Again, I don't know if you've seen the latest on even taking the vaccine, John. So, but they'll find 100 people, I think. And should be interesting to see. And again, let's move on, right, John? We'll go to the next topic.
Sure, absolutely. We'll watch it, and I'm sure we'll hear more about it as they get closer to the study kicking off.
Because I think we could talk the whole podcast on this, and it would be fantastic. It would be fun. The next one's a little bit, I don't know if controversial is the right word, but who decides when vaccine studies are done, right? This is internal documents show Dr. Fauci plays a key role, which On the surface, to me, I didn't dig too much in the article, but somebody has to, John, right? So tell us a little bit about the article.
So I dug into the article. The headline says a lot, says something, but the article says more that is slightly contradictory. It's basically the involvement of Dr. Fauci is there because he heads NIAID, but many of the leaked vaccine candidates as funding from the US government as part of this Operation Warp Speed. And so this has meant a different structure to the Data Safety Monitoring Board that you would have for any trial. In these cases, because there's government money behind it, there's actually NIAID are able to nominate people to be in those data safety monitoring boards. Another piece of it is the membership for all these vaccine candidates. There's commonality, so it's the same people looking at the J&J vaccine, looking at the AstraZeneca vaccine, looking at the other vaccines coming through. There's some benefit to that. There's one vaccine shows some adverse events, then for that data safety monitoring board group, they don't have to learn about it when looking at another vaccine, but they can certainly look at the results from those other vaccine candidates to see if there's any potential for the same thing occurring. I think the involvement of Dr. Fauci is if there's sort of conflict between the data safety monitoring board's conclusions and the company is the one who makes the ultimate decision, but having Fauci's name associated with it actually gives probably more weight to DSMB, so there's unlikely to be pushback is some of the assessment of his presence here. So I think it's like, yes, his name's involved, but ultimately he's not the one driving the decisions. It really is independent people who are on the board, and they're doing the job that they would typically do for any vaccine candidate.
You nailed it. That's the take-home message. It's being run accordingly, despite the speed of it. And yeah, there's going to be some what I think of as like commingling, but good people do good things. And at this point, there's no evidence of true biasness. There's hearsay, there's finger pointing, but nothing is jumping out that's abnormal in the whole process. There's always going to be overlap. And at this point, I think we would agree, John, we've talked about how much more collaboration's going on. His oversight, I think, is net neutral. It's like having the vice president over the Senate, that kind of oversight.
Yeah, exactly. The only exception to this is the Pfizer drug is not part of this because they've looked to fund it completely independently. So it's going to be a different board looking at their data. So I don't view it as a huge deal at all. It's interesting. And it just, like I said, I think Fauci's name will give some credibility, but actually it doesn't really change the process at all.
That's the point of why we found it important enough to make our podcast. So, our first non-COVID news: ex-doctor was a threat to millions after faking Tri-City medical drug trials. I mean, this is like straight out of a Hollywood script, John, right?
Yep, we try and talk about the positive things that are happening in terms of COVID and general drug development, but there are some unsavory characters out there involved in this business, though thankfully they're in a minority. And this was a guy who ran a clinical trials organization, was sentenced to 28 years in federal prison for falsifying clinical trial data. But not only did he fake data, but he used threats, intimidation, and retaliation to hide crimes. He intimidated his employees. He threatened them at their homes, at their place of work, slashed their tires and stalked them in order to prevent them from cooperating with authorities. I mean, there's talk of him flushing drugs down drains and not actually administering them, fake electrocardiograms and vital signs, made up subject diary entries. So all things that are vital to the conduct of a clinical trial. He was doing whatever he felt like.
That's the word. It's a deplorable act by this individual. Yeah.
Yep, we try and talk about the positive things that are happening in terms of COVID and general drug development, but there are some unsavory characters out there involved in this business, though thankfully they're in a minority. And this was a guy who ran a clinical trials organization, was sentenced to 28 years in federal prison for falsifying clinical trial data. But not only did he fake data, but he used threats, intimidation, and retaliation to hide crimes. He intimidated his employees. He threatened them at their homes, at their place of work, slashed their tires and stalked them in order to prevent them from cooperating with authorities. I mean, there's talk of him flushing drugs down drains and not actually administering them, fake electrocardiograms and vital signs, made up subject diary entries. So all things that are vital to the conduct of a clinical trial. He was doing whatever he felt like. And the thing, and it Oh, it's awful, and it has a real consequence on people because I mean, there's one 3-year-old girl was permanently scarred after being enrolled for a scabies study or when she suffered from eczema, and then there was another teen girl. I was, yeah, yeah. Sorry, I missed this one. Subject died while on 2 trials at once. He wasn't monitored, and his death was never reported. I mean, you have to be reporting adverse events like that. And then another one was a teen girl tried to commit suicide while on smoke cessation trial, where the company scientists were already concerned about that suicide as a potential side effect of that drug. So, yeah, just awful, awful situation, and he thoroughly deserving of his 28 years in prison.
To me, this is one of the worst stories that I've read related to falsifying data. This isn't some individual getting their PhD or maybe skewing things left and right so that a bioanalytical method passed and had maybe minimal consequences on any sort of drugs. I don't know if this individual thought he was Tony Soprano or something, but this is just disgusting, right?
Oh, absolutely. Yeah. The overall assessment was provided false clinical trial data to drug companies and FDA, so affected safety data on dozens of drugs designed to address heart disease, diabetes, asthma, pediatric illnesses, adolescent smoking, cirrhosis, scabies, depression, opioid addiction, and other indications. A wide range of medications that people need, but they need to know they're safe. And yeah, it's just awful.
Let's move on to something really cool coming out of MIT. Universal flow vaccine by refocusing the immune system.
Yes, I mean, basically, the flu is something that mutates pretty rapidly. So many of the vaccines are really they're attacking that region that is the one that shifts. There's a much more stable region, and so the aim of this group is to really try and design a vaccine to get into the stable region, which is less accessible, and preclude the need for having a fresh vaccine every year.
John, are we ready to go into our exciting uh, 25th anniversary podcast?
Yes, let's move on.
Today's podcast is we're going to share a little bit more about ourselves and how we got here. And John, I've been thinking about this the last few days about where to start and how we got to where we are, but I'm going to let you go first.
So we thought we'd talk about experience, what our backgrounds were, and what pulled us towards drug development. Obviously, our two backgrounds are very different, and I'm going to start off with the UK, and I'm actually going to go pre-university because the UK education system is very different here. You specialise very early, and actually you make a decision at age 14 that really sets your path for the rest of your life if you stick with those decisions. What you do in both in Scotland and England. Scotland, you do what are called O grades, then higher, and then you go to university. England, you do O levels, and when I was at school, did A levels, and actually, I did the path of doing O grades, higher's, and then A levels because I wanted to go to university in England because my family were English. Where I'm coming from, at age 14, you have to decide what 7 subjects or 8 subjects you're going to do for O grade. And you basically, maths and English are compulsory, but then you make that split between arts and science. So I was obviously going to be scientific because I haven't got an artistic bone in my body. In Scotland, you couldn't do physics and biology. To my eternal regret now, yeah, this is why I struggled to keep up with you, and it's why I raised it, why I'm going so far back. This is why I struggled to keep up with you. I went with the way of physics because I listened to my dad on this one, and I should never have done. I should have gone biology because it interested me. I was always going to do chemistry because I had that interest in that, and the biology chemistry intersection would be much more valuable to me going through life, and actually would prove to be much more valuable now. But that is sadly a gap in my education. But I'm going to pass it back to you.
John, I had a similar experience growing up in terms of getting advice or at least making decisions, but and that was I was actually an international relations major when I started at Stonehill for my BS degree and quickly switched to pre-med. But let me even go a small step back. What in high school, I was the guy who took a bunch of AP courses and just because they were easy. Literally, I didn't find high school challenging at all. And teachers were pretty much like, you need to be taking these courses. And it was in part because I just was fortunate enough to have good memorization, John. And that carried me Very well. I mean, I wasn't the smartest kid. I probably played around a little too much to be graduating as valedictorian. I had fun in high school. So I do get an academic scholarship to go to a place called Stonehill College. As I mentioned, started as was going to go into because I spent a summer in Spain, and that was in between my junior year and senior year, and I think that really helped shape me. So when you go abroad by yourself, you grow up real fast, and then you have to learn a second language, and you realize there's a big world out there. So what happens towards the end of my senior year, I'm really doing well in AP Physics and AP Biology, and suddenly I'm like, I think I got a calling there. So I jump into pre-med. I do pretty well. I graduate with the 3.5 or something, and it's not hard. I have a fun time, but I still don't really know what I want to do. Other than I spent a lot of time dissecting animals, and I knew I didn't want to do that. So John, I can carve an animal up like nobody's business, because I was a tutor for that for a little while as well. Worked in the lab for 2 years as a student teacher. And so I get a internship at a company called Cellcore, and the rest is kind of history. That company, I learned about autologous T cell therapies for metastatic renal cell carcinoma patients, and the passion in me is just triggered. And so that was basically what is now CAR T cell stuff. So I go there and I start meeting people. I didn't even really understand graduate school. I was really funny. My parents were. My dad was a military guy. My brother graduated college. But he was not going to go to grad school. So somebody says to me, I get a job, and then grad school comes on, and I get a degree in biotech and biochemistry from the University of Massachusetts at Lowell. And then I go on and get my doctorate from the University of Pittsburgh School of Medicine. But all along the way, from the moment I'm a senior in Stonehill College, I do an internship at Cellcore. And throughout my master's degree, I work at Cellcore for a good 5-6 years, John. So that sets the foundation for my immunology passion. And I'll pass it back to you, and then I'll talk about my PhD and so forth. And maybe I do a nice master's degree too, but I'll talk about that in a minute.
Yeah, I think that whole piece of doing industrial experience. I know you were doing it independently, but I think degrees where you're set up to do an industrial year or a year out really helps in terms of preparing you for the workplace. My son did that with his communications degree from Drexel. It was a 5-year degree, but structured so he worked through it, and I think it really does help help you mature more. And I will touch on this as regards how it directly affected me. I did not take that approach. So anyway, going on to university, like you said about the coasting through school, I coasted through school as well because I was a bright kid. It wasn't a challenge for me. So I think going into university was a bit of a shock. I did not apply myself as I should for my bachelor's degree. And in the UK, your degrees are graded, so you get a first 2-1, 2-2, third pass or fail. My background is my dad actually was a lecturer at University of Strathclyde in Glasgow. I was always looking at going that path of doing a PhD and going on from there. So when it came to the bachelor's, I actually came out with a third, which is one of those things that basically says you're not cut out for this, time to leave and find something else to do. And actually, it was fortuitous. I'm a great believer in serendipity and how a single thing can happen that really helps the rest of your life. If I got a good degree from University of Reading where I went, I would probably have done a PhD in inorganic chemistry. Which looking back in it was a really poor fit, but I had a professor who wanted to work with me. Getting a poor degree that closed that door. So there I was. I ended bachelor's. What do I do now? And I actually was looking in one of a weekly scientific magazine, and there was a small ad for a master's course at University of Bristol for advanced analytical chemistry. I thought that could be interesting. I quite like the sound of that. So my parents actually paid for me to do that one, which it was a year course, I think 9-month taught, 3-month research. When it came to the research project, the professor basically distributed projects to people, and I was given this project to look at oxalinic acid in water. And oxalinic acid, if I remember correctly, was an antibiotic administered, in this case, to fish, and that's why they wanted to measure it in water. Looking at the structure molecule, it should really have been an LC project, but other people had been allocated the LCs before I got even allocated my project to work on. So I was given gas chromatography to work with. And this molecule was not GC friendly. So the bulk of it was actually then spent looking at ways to derivatize oxalic acid to get it in a friendly form to then run by GC. And then right at the tail end of the project, he'd got independent funding. And so it was the local water board. We were allowed to visit and actually run, not just with GC, but run my extracts into GCMS. So this is my first real hands-on chance to touch mass spectrometry. And it was like a light bulb going off where it was like, I really enjoyed the separation piece of chromatography. But when you could actually had something at the tail end that could tell you what your peak was, it was like, this is fantastic. And this is where I want to go from here on in. So that was the masters. When it came to PhDs, I was still intent on doing a PhD. I couldn't do anything that was government funded because my first degree was so poor. Luckily, at the year I was applying, there were 2 PhDs that were industrially funded, so there was no government money at all. I messed up the interview for the first one at Bradford, but luckily came across my guardian angel, a guy called Di Games, who specialised in taking people who maybe needed picking up, dusting off, and setting on the right path. And he accepted me to do a PhD at Cardiff, and that was the life changing experience to really set me on.
John, hey, I'll so we've got a similar sort of pathways there. I'm at Cellcore working as a cell processing engineer one in a class 10,000 clean room, which is exposure to many, many things. But the God's honest truth is I was just a good set of hands and had nice endurance because you had to stay in that room for long periods of time. But I didn't know what the heck I was doing. I knew I was treating metastatic renal cell carcinoma patients. And I say that with such eloquency because I didn't really know what the hell it meant, right? This is going back to like 1994. I had done an internship there looking at the efficacy of different disinfectants within the clean room and the areas like the tabletop, stainless steel, and wall. And I used to have to sit there and like put a known quantity of bug on it, spray it with each of the various cleaners, and then take what was called a Rodak plate and see how much was there. So I was doing microbiology and I didn't like it, but I loved the company and they were doing all this cool stuff. And they said, well, you seem bright enough. And so they hire me. And so I get to do that cell processing engineer job. I'm doing that. I'm really enjoying it. And I meet one of my first mentors. His name was Bruce Babbitt. And that's when he's a PhD. I'm like, what the heck's a PhD? That's how ignorant I was of graduate schools and things like that. But I'm like, oh. And so I just start, he kind of mentors me along. And so rather than jump into a PhD, I needed to beef up my background on dendritic cells and T cells. So I went and got a master's degree from UMass Lowell while working at Cellcore. But I did take a pause to about 10 months off to do all the bulk of the classwork. And then I did an internship, which would then be my second internship at Cellcore. And that's where I do my thesis degree on human anti-mouse antibody responses, which are HAMA responses, which is now immunogenicity or anti-drug antibody responses. So I date back a long way in that type of stuff. So that I do my bench work at Cellcore. I get the master's degree, so that's great. But now I am thirsting for more education, and I find my way at Pittsburgh through, again, some nice, hey, this school is doing great immunology work, so I go there. So that was for me, I hope that shared with you a little bit of how I got to where I was. 2 internships, a lot of like finding out where, like I mentioned, Bruce Babbitt, there were some others, but I was like, what? I think I want to get into the research side. And then I'll share, when I finished my master's degree, I had a crossroad. I had gotten accepted into a PhD program, and I actually had looked at patent law, and I was very close to going there. Didn't, and I don't think I regret it, John. How's that? I don't know. I don't, because I think patent law. I think I have big aspirations as I get older, and someday I may still get that, just for to put more letters at the end of my name. But I don't regret it, and went on to get my doctorate at the University of Pittsburgh in immunology. But I'll let you share a little bit more, and then I'll tell you about some of my fun times at Pittsburgh.
Yeah, so the timing of me doing my PhD was at a time where liquid chromatography mass spec was not routine. I mean, our business or at least part of the business, is based on LCMS, but the technology now is such that the bulk of the work is done overnight where no one's in the building, because we figured out how to interface liquid chromatography with mass spec. We now have a team way of doing it. Back when I started my PhD, the problem with LCMS, and I know I've touched on this before, is LC is a high pressure technique, mass spec's a high vacuum technique, and to combine the two, you need to find a way of interfacing two supposedly incompatible technologies. So with Dye, he'd been doing LC. He was actually one of the people who designed a moving belt interface, which is basically the outlet from the LC would deposit on a belt that was heated, and then by the time it reached the source of the mass spectrometer, any solvents would be evaporated, and then then you'd ionize what was left on the belt into the mass spectrometer and analyze it that way. So he'd been interested in LC. He actually got interested in supercritical fluid chromatography because that would mainly using supercritical carbon dioxide with dopants like methanol or 2-methoxyethanol in small proportions. And so the thought was that it would actually be at least a bit more compatible with the mass spec than full-on LC, because it's easier to volatilize at least the carbon dioxide portion of it. So I did that. So he got interested in it. There was a lot of interest in industry because, again, we didn't have a routine way of doing LCMS. So we had a constant flow of visitors. You got very good in terms of connections. To cut a long story short, I got to end my PhD, went to a conference in Bordeaux while looking for a job, which is a fantastic city. I'd recommend you visit it anytime. And actually saw an ad for a postdoc in North Carolina.
I want to touch on the platform. So I like the way you talked about how you like and how relevant they are to our drug development careers, right? As an undergrad, it was all dissections and I did some chromatography work and I hated it with the glass and oh God. And so then my master's degree, I was doing ELISAs, a lot of Western blots, some PCR. And then at CellCore in particular, I learned 4-color flow cytometry and tritiated thymidine cell proliferation assays, which are still, they're fading away, but what a powerful tool that was at the time. And then at Pitt, I learned to have more colors in flow. I go on to learn ELISpots. I do a rotation on a Biopharmaceutical system, and I do a rotation in an LCMS lab. So when I joined Pitt, I was in an interdisciplinary program, and you got the opportunity to do 3 rotations. I chose to go ahead of class and rotate in Walt Storkis's lab, where I ended up getting my degree, and it's a heavy immunology laboratory, excuse me. And so I said, I then go into a base. I do rotate. I still take advantage of my other rotations. I go a little bit backwards into a more heavy academic laboratory for a rotation. And that gets some more meats and potatoes around some real cool ways of looking at T cells using different methodologies, like a given cell line to some proliferation assay, HT2 cell lines, they're called, for proliferation assays and responses, just to get to me, I think of it as like the blocking and tackling. And this is at the advice of Walt. He's like, go into this lab because you can learn, a little bit more fundamental and foundation of true immunology research. So it was great. And then I go and he's like, do whatever you want. And I go into a Biocor LCMS lab with a guy I play basketball with, just I kind of befriended him. And he's like, oh, come rotate my lab. I'm like, sure. He's in like the biochemistry department, but it was good stuff, right? So Biocors give good understanding of on-off rates of proteins. And then obviously I see the mass spec and I'm like, oh God, not for me. I knew that was a little bit, it was just, I didn't have the chemistry background, John, at the time. And I'm very excited to get back into my immunology lab. So that gives a little flavor of the platforms that I'm learning. And then all along the way, one of the things, you mentioned it, there's some great people who come and speak at Pitt, and that I took advantage of all of that. I was in the lab that I joined had 6 people in it. By the time I graduated, it had about 18. It was fantastic, and I got to know the university really well. And even today, I still have some networks from there. So, John, I'll take a deep breath and let you jump in. But that's going to bring me up to Pitt. I hit a crossroads when I'm graduating with my immunology degree, and I'll share with you a little funny story around the Gardasil vaccine. But go ahead and.
Sure, absolutely. Yep. So back to Bordeaux, where I was actually there with my then girlfriend, who is now my wife, and that's thus the 32nd wedding anniversary. Basically, I applied for this postdoc in North Carolina, and I got a phone call a couple of weeks later saying, "Well, when do you want to start?" I'm like, what? So I think I was probably the only applicant. And so my wife and I said, well, that's it. We're going to go and do North Carolina. We got our visas, got some stuff shipped over, and spent 3 years living in Raleigh and working at the NIHS in Research Triangle. And the guy who ran that lab was a guy called Ken Tomer. Ken, again, it was all LCMS based, but he was looking still at the stage where we don't have a routine interface between LC and the mass spec. And so he was looking at chromatography, the very 75 micron ID LC, and they were looking at fast atom bombardment was one of the ionization techniques they were looking at, where they basically, and that relies on glycerol to act as a matrix to then fire fast atoms at the end of the probe that was going into the mass spec, and that they would help ionization of the species that you're looking at. It was a real art to what a girl called Lisa Dieterding and Art Mosley were doing, where they'd actually thread their chromatographic column down through the middle of another capillary, the glycerol be delivered coaxially. And like I said, there's a huge art to doing that. So I wasn't allowed near the fast atom bombardment piece, but actually an electrospray source was delivered onto one of their small single quadrupole. And this electrospray, as we've mentioned in multiple podcasts, is the key to why LCMS is now a routine technology, and there's so many labs providing the services to analyze drugs and therapeutics to support drug development. I was just looking at, small molecules. And then another piece of the separation part that Ken was interested in was actually capillary electrophoresis. And one of the supposed selling points for capillary electrophoresis was that you could look at separate very complex mixtures. So one day in the NIHS library, I was looking through a book and stumbled across a book about snake venoms. And luckily, partway through the book, there was actually a fold-out page with various sequences listed. And I'm looking at these sequences, figuring out the molecular weight, and figured out there was enough lysines, arginines, and histidines that those protein polypeptides should come into the mass range of the mass spectrometer. So I was thinking that would be a really good thing to look at CEMS of snake venom. So I actually ordered a bunch of venoms from Sigma, and that was then my life for the next year. It was absolutely fascinating. I loved it to death, and doing all these sort of things. And there's so much more I could have done with snake venoms, and sadly, my postdoc came to an end, and I then had to go back to the UK because it was a condition of the postdoc that you had to go back to your country of origin for 2 years, at least 2 years. And so I'll be very quick on this. Went to a company called Kratos Analytical, but it had just been bought by Shimadzu. And Kratos were known for large mass specs, but were just launching a time of flight mass spectrometer. And so I went in the door on my first day expecting to do one job and immediately got diverted to doing another. But again, it's serendipity. It pushes you in a direction you're not necessarily thinking that's what you like to do, but it actually sets up for success later in life. So I actually ended up looking for applications for this time of flight mass spec, worked with marketing to produce marketing materials, interacted with customers. Actually, the time of flight had been designed such that it was relatively portable for a mass spec. So we'd put it in the back of a car and drive to customer sites and run samples. And so I actually did a driving tour around Denmark and Sweden, doing that with a mass spec in the back of the car. You'd set up in the lab at the customer's site overnight, pump it down, run samples the next day, put the mass spec back in the car onto the next site. So that was actually my first real sort of interaction with customers and starting to develop that how to work with customers. Working with marketing was great because I had the data. So it was actually working directly with someone who could bounce ideas off each other, how to present the data, how to sell to really show what your instrument could do for you if you have these kind of issues. And funnily enough, snake venoms by time of flight was one of our marketing materials. So again, it keeps coming back to the things but you can use it as a selling point. So that was Kratos. Sadly, with the big mass spectrometers they were known for were making no money, so they decided to stop producing them, and that meant a reduction of workforce from around 200 to around 60 people, and that was where I then left, which then set in motion me coming over to the US. And I'll pass it back to you, and then I'll come back to that.
Yeah. I think we're gonna have to save. So, John, you're gonna have to. That's job one out of your postdoc. You're gonna, and on the 50th podcast, you'll have to pick up with your next job because I think we're just running long. So I'll wrap up quickly with. So as I mentioned, I've got 3 degrees. I worked at Cellcore. I didn't mention Cellcore actually. We kind of show up one day and the doors are locked. And I had been working there the better part of, I don't know, close to 6 or 7 years or something. So what happens is a lot of us, Bruce Babbitt lands at a company called Allegix, and a lot of us he's consultant for him, and a lot of us get recruited to go work there. And so about 6 of us from Cellcore, and we were only 30 people at the time, wind up in this company called Allegix. That's where I meet my wife. And so I do a 9 or 10 month stint there. And then I go to Pittsburgh and she follows me a year later. So I get out of Pittsburgh. I'm at not a crossroad. I have opportunities. I did my whole doctorate on the human papillomavirus. I worked hard with the McGee Women's Hospital there in Pittsburgh. I got over 100 women to donate 100 mils of blood, in addition, a piece of their cervical tissue, in conjunction with a couple physicians and some fellows up there. One in particular, Mira was her name. She was great to work with. She went on to get an award. I got honored in the McGee Women's Hospital as a co-presenter with her at that award show, which was in grad school when I was a little bit older, maybe 28 at the time. When you're looking at somebody who's like 22, they're like, how did you get up in front of 30 physicians and give a small, brief presentation? I was like, I don't know. But when you put it that way, it's scary, isn't it? It just happened, right? So somewhere in there, I'm doing some cool stuff. And the degree that I get is it's all about the papillomavirus responses. And we demonstrate that early onset of what's called cervical intraneoplasia. Women's immune responses are X towards the papillomavirus. As they get into the later stages of that SIN 1, 2, they get into SINs 2s and 3 stage, their immune system's Y. And then as they get into full-blown cervical cancer, the immune system is expressing itself Z. And all of that's done through ELISpot method for their T cell responses against it. And so their immune system goes from I studied helper responses, 4 responses. They go from IFN gamma producing to more of this T helper 0 is how we define it, where they could produce a little bit of IL-5 along with some were making interferon gamma, which is a sign of the immune system losing. And then we go into this full blown, they're making IL-5 and no interferon gamma anymore, which means they've gone from acute to chronic. And that's all the rage. It still is. That's that acute inflammation you talk about. And so Merck gets a hold of this and somebody says, hey, we want you to come work on Gardasil. And I'm like, no. And the recruiter calls me back and says, Dr. So-and-so, the vice president says, hey, you really should hear him out. And I say, no, I'm not moving to Philly. My wife and I are looking for opportunities in the Midwest. So the dude called me and he's like, son, I don't understand why you don't want to come work for me. And I'm like, look, I really do, but I got this beautiful wife of mine over here. And so I go to Streck Laboratories. And John, I'm going to stop there because I had a wonderful time at Streck and it was I don't have any regrets, although one of our first weeks in Omaha, my wife is looking at the Gardasil commercial and she's like, you could have gone and worked on that, right? I'm like, yeah, it was like a recruitment for it, I think. But anyhow, John, we're right up against like an hour time.
I can wrap up quickly. We talk about my 26 years in bioanalysis. Well, 24 was in the next stage. So I will do it quickly though. Come out of several months unemployed, we got to a stage where it was choose between pay the mortgage or fund me to go out to ASMS to look for a job out in the US because there wasn't much happening in the UK. Well, there I talked to Jack Henyon, who coincidentally used to teach LCMS courses with Die Games. So you got the old boys network working on my behalf too. And so I ended up working for Jack at a company called Advanced Bioanalytical Services, a CRO providing quantitative LCMS. So this is my chance to actually do liquid chromatography on a conventional scale, because I've never seen a 2.1 millimeter ID column before. I'm used to working in 75 micron. But I learned a ton there. I started as a bench scientist doing method development, running samples, validation, ran the instrument, extracted samples, did everything. Then eventually took on the responsibility. My teams were responsible for all validation and sample analysis, and just learned a lot. Learned again, built on my customer interaction experience, and ultimately getting your hands in projects over a 24-year spell while I was there. You see so much, and it really helps then troubleshoot issues when you come across them. Now, the other quick highlights: we actually bought a lab in the Netherlands, which I ended up running for 18 months. That was fantastic because it was that opportunity to live in the Netherlands. At least professionally, it was fantastic. Personally, it was a little bit weird because I spent so little time here and I was jet lagged all the time. Also helped set up a lab in Beijing. Finished with Q squared as they were then, and joined KCS last October, and the rest is history.
Yeah. Well, I guess I can go real fast. I go to Streck for about 3.5 years. While I'm there, working on 510K clearance of a preservative tube for the T cell, B cell, NK panel. But what I learned a lot there in terms of just, like you said, I begin to get more client interaction. I'm traveling and presenting posters and working with a lot of large pharma. In fact, BD Biosciences ends up private labeling this reagent as well for combating the HIV initiative, and it's still used quite widely in clinical trials. Omaha becomes small for me, so I start to look for other jobs. My brother actually came up, and I was offered a chief scientific officer role for a small biotech in Boston. But my wife finds a job at a company called IBT Laboratories, and it reads like my resume. So we moved to Kansas City. It was great, and I won't. But it was all about Ellie. They were looking for flow and Ellie spot, and I was like, "Oh my god, this is like everything I do." And I didn't really understand what outsourcing was until that job. They were a reference laboratory. So my first salvo into the CRO space is in the reference lab space, but I'm working in the biopharmaceutical services, senior scientist and director of that. And all along the way, the BD reps are like taking me, I'm traveling about 50% of the time at times and getting gift cards because I'm helping get their kids through college with how well we're growing the business. And all along the way, I'm still somewhat in the lab, John. And it was great, but that did that for 6 years. And the company had grown from IBT to ViraCore IBT, and now it's a Eurofins site. And then joined KCAS, and that tallies about 15 years in the CRO space. And if you count the 6 or 7 in Boston in my academic years, it's really about 26 years, all in like immunology. And the drug development component was heavy at Cellcore, obviously not so much at Pitt. Streck was because of the clientele we were trying to service with the tube. And then obviously, as soon as I get into IBT space, they're working with some pretty big allergy and immunology pharmaceutical companies. So that's where I learned the mesoskill discovery system, learn a lot about immunology and allergy, which I had known a little bit about. And then, coming to KCAS, I was then into the business development role, but always technical. So, John, we're right up against a good amount of time. I think I don't know if we have any feedback, but John, that was, I had a good time.
I'm just going to say something very quickly. I think it's interesting that we both have that, and it really plays into the role here, where we're in the lab, but the enjoyment of going out on the road with business development people and interacting with customers is huge. And not many people, in my experience, really want to do that. I always enjoyed that blend of roles and duties, and that's why I enjoy doing this now.
Yeah. It just seemed natural for me. I didn't even really understand what I was doing, but I just went in and was able to kind of just surprisingly listen to people and quickly understand, and had a lot of just like, oh, I've touched, I've done this, or I've seen that. And like you said, it just builds and builds. And now, I mean, the number of drugs that I've worked on is because of co-meds and things like that. It's really high, right? I mean, I don't know how many drugs you think you've touched, but at KCS alone, I've done at least 100 biologics, and that's just, never mind the probably 20 small molecules attached to the biologics, right? So that's all been great stuff.
Yep. For a long time, I was the not really business development, but was always the public face of Advia in Europe and laterally, Japan, because I was willing to travel and combine it with other things. So yeah, I always liked doing that.
Yeah. So, John, I think we can move on to some feedback. And by the way, congratulations on 25 podcasts. I think that was a lot of fun. Enjoyed getting to know you a little bit more. You are definitely nerdy when you start talking about mass specs, which is great. Your passion runs strong. I hope you hear it in me when I talk about some of my vaccine experience or even immunology experience. Jeremy actually wanted to be the first one to give us some feedback. He says, congratulations on 25 episodes, guys. I couldn't ask for a better duo to record and edit every week. Well done. Well, Jeremy, thank you and for all you do.
Yeah, thanks very much, Jeremy. Despite my suspicion about the podcast when we first raised the idea, this has been a lot of fun. I'm certainly looking forward to 25 more episodes, so quite sure what we'll do for the 50th. But yes, it's been great. Also, feedback. Erin, our co-producer, who's back from maternity leave. Very nice to have her back here. Wow, 25 episodes. Congrats. I love the quick snapshots every week of what's happening in the drug development world, especially during COVID times. Nice job, guys. Thanks, Erin. I know I've alluded to this in the past. It's really nice to do this podcast because it forces us to some extent to actually look into the news and find out what's going on. So I've learned a ton just by doing this. And yeah, really, really, really do appreciate.
John, that's a good point. It does, to some degree, force us to try and stay relevant, which we should. That's part of our job, to be up on technologies and what's coming down. But you're right. It's always enjoyable. It makes it a little bit better. I'd always done it, but now I do it with a little bit more purpose and a little bit more dedication. And I really do think it's helping just in general keep you up to date on a lot of moving parts, a lot of moving industries, but it's a lot of fun to cover it from a COVID and non-COVID perspective, right? So, John, I think that'll do it for this one. We can talk a little bit about podcast number 26. That's going to be picking up on our non-GLP, non-clinical series. It'll be part 3 of 3. And John and I will wrap up on non-GLP, non-clinical and how it flows into preclinical. So we'll give a little bit of a summation of the non-GLP, non-clinical space and then how it helps really with the drug development process into preclinical. John, exciting topic next week.
Should be good. Absolutely. Yep. Looking forward to it.